Hi biologists, let's start by looking at the learning objectives for this lesson. Take note that this material is for the higher level biology paper only. At the end of this section and following the biology syllabus, you should be able to explain the term optimum activity as applied to pH range. Describe the heat denaturation of protein. What does this actually mean? What are we trying to understand? Well, you have to be able to explain the term optimum activity and understand how pH range or different pH values affect optimum activity. You also have to be able to describe the heat denaturation of protein, or in other words, how heat affects protein. As we know already, enzyme activity depends upon 1. The active site and 2. A suitable environment. Why is this the case? Well, don't forget, enzymes are three-dimensional globular proteins. Therefore, the active site has a three-dimensional or 3D definite shape. And the shape of the active site is specific, meaning the enzyme will be complementary or match just one substrate. Enzyme activity is affected by the environment in which they are found because enzymes are proteins and proteins are affected by environmental pH and temperature. What is meant by optimum activity? Optimum activity is an important idea, so again we have to make sure that we know what this term means and that we are able to explain it. Optimum activity is referring to the idea that the rate of enzyme reaction, the rate of an enzyme controlled reaction, is happening at a maximum. All enzymes, don't forget, have a specific pH or an optimum pH or a pH at which they work best. Optimum pH. The optimum or the ideal pH for most enzymes is a pH of 7. In other words, most enzymes will work best in neutral conditions. The rate of reaction is at a maximum in these conditions because all the enzymes are in their native state. In other words, they're fully intact, they are in their original shape and structure, and the active sites are all fully intact. Optimum pH will obviously depend on where the enzyme works. As we've said, most enzymes work in neutral conditions at a pH of 7. But pepsin in the stomach, pepsin, don't forget, is a catabolic enzyme. It breaks down proteins into peptides. It is found in the stomach and it works best at a pH of 2 due to the fact that there is hydrochloric acid, stomach acid, present in your stomach, which creates an environment of pH 2. How does pH affect optimum activity? Well, as we've learnt already, small changes in pH can affect an enzyme's activity. And any change in pH can cause the rate of activity to fall. Why is this the case? Well, this is because active site shape changes and is no longer complementary to the substrate. Extreme changes in pH causes the enzymes to actually denature, to change their shape completely. The shape of the active site and the shape of the enzyme is so changed that they permanently lose their function. They can no longer catalyze chemical reactions. This heat denaturation of protein is a very important idea, and we must realise that when most proteins are heated above 40 degrees Celsius, they lose their 3D globular shape. Remember, their 3D globular shape refers to the idea that proteins are folded. When they are heated, they unfold or they lose their shape completely. They are so changed that they can no longer function. Enzyme denaturation is a definition that they are very fond of in exams, so make it your business to learn it. Denaturation is where an enzyme permanently changes shape. We have to have the idea of permanently in there. There's no going back on this. Once it has been heated and has been denatured, it has changed its shape completely, and this is a permanent state of affairs. Because these enzymes are found in cells and carry out biological activities and catalyze biological reactions, we must include the idea that it's going to lose its biological activity. Denaturation is caused by high temperatures, as we've just said, pH values outside the optimum pH, and some chemicals or radiation can cause denaturation. So in a nutshell, what is the effect of pH change and high temperatures on enzymes? Well, it's simple enough. The active site shape irreversibly changes. The substrate can no longer bind or combine with the enzyme. The enzyme is denatured and no longer works. And there you have it. Now that we have reached the end of our lesson, have we achieved our objective? Can you explain the term optimum activity as applied to pH range? Can you describe the heat denaturation of protein?